So the last part of this chapter has to do with recombinant DNA technology and the applications that we have. And so once we have a cloned gene, one of the things that we most often use it to do is to produce overproducers. It's a way that we can accumulate lots of different types of proteins in large quantities. And a lot of times we can then use these. If you look at this table below, these proteins that are produced by genetic engineering, we can use these then as treatments for many different types of illnesses. Other things that we're able to do once we have our DNA of interest in an expression vector, we can put that vector with DNA that allows us to turn on expression of the vector under certain environmental conditions or turn it off. And so that also allows us to place our gene of interest into an organism and then control when that organism would be expressing the gene or not. And that's very, very helpful in designing some types of experiments. For instance, if you have a yeast cell that has a vector in it and it only expresses that vector at a high temperature, you can grow your yeast cells at a normal temperature for them, and when you switch it over to the high temperature, they'll start to express your protein of interest. So by controlling the conditions at what time by controlling the conditions that you put the yeast in, it then allows you a time control as to when your cells are going to express your protein of interest. And lastly, we can place our expression vectors in eukaryotic organisms. So being within a eukaryotic organism, it allows these proteins to also have the modifications that you might expect if they were in a human cell. One of the other ways that we can manipulate DNA is actually by mutating it. And so we study genes and proteins, and we know that the central dogma of biology is that DNA is transcribed into messenger RNA, and that's translated into a protein. So if you want to flesh out and figure out the functions of proteins, sometimes we're able to mutate the original DNA, and then we can affect the structure of the protein in the end. We like to study mutated proteins then because it helps us to determine the protein's function. We can identify binding partners between different proteins. If a protein is an enzyme, we can find the active site and how mutations might affect enzymatic activity, etc. And so how we make a mutated gene is actually fairly simple once we understand how PCR works. And so if we look at this picture, this green circle here, this is not a plasmid. This whole thing is a gene of interest. And they're showing us the nucleotides for this area. This area is of interest and we want to mutate it. So what we do is we get an oligonucleotide. So an oligonucleotide is generally a sequence of nucleotides that's between 10 to 20 nucleotides long. And we can mismatch nucleotides in here. So we can mutate this sequence via PCR. We can change the nucleotide sequence in it. So here you can see is our mismatched oligonucleotide. And then we can do PCR with this oligonucleotide. There's enough nucleotide similarity to this sequence of interest that the oligonucleotide will bind, but you can see we have these mismatched nucleotides. So when this primer anneals and then is replicated, you can see we replicate the gene identically. It's all similar except where we have these substituted mismatched nucleotides. So then when we have this structure DNyl, our new gene, what's in purple here, is going to have a different nucleotide sequence. And so the different nucleotide sequence will ultimately make a different protein. The protein will have a different structure. And if you change the structure of the protein, odds are you will change its function. And now we'll be able to study what perhaps this enzyme, or if it's not an enzyme, this protein, what its function is in a cell. And by using this technology, we have fleshed out the functions of proteins, and we've also been able to mimic mutations in DNA that have caused diseases. It's helped us to understand how a mutated protein and particular diseases affect the patient. We've also branched into making genetically modified organisms, or what we call GMOs. And I think a lot of people are familiar with these just from walking down the grocery aisles. You'll see a lot of products are now labeled GMO free. That means they are genetically modified organism free. There is a debate and controversy around putting GMOs into the food chain, because now you understand how genetically modified organisms are made. It all comes down to the DNA, and you've seen that we have to incorporate plasmids into bacteria to be able to make these genes and manipulate these genes. And so there are many groups that are worried that the methodologies that we use can affect people because we are playing around with antibiotics and antibiotic-resistant genes. And so there is some degree of uncertainty with health effects. However, in the United States, this is still a big debate. You can see in this list that we use genetically modified organisms in a lot of different areas, like agriculture, definitely research. I put recreation on here. You can go to Walmart and buy genetically modified fish that glow in different colors now, and that's all because of DNA manipulation. And there's many medications. So EPO is erythropoietin. That induces proliferation of red blood cells. Many people are familiar with insulin. It's used to treat hyperglycemia. TPA is breaks down blood clots, and interferons are given to help thwart the activity of certain viruses like hepatitis in the body. When we make genetically modified organisms in the laboratory, we can use different types of manipulation to see different things. And so a couple things that we use, we can create what we call transgenic organisms. Transgenic organisms is when we insert a DNA sequence of interest into a specific area on an organism's chromosome. And we can create what we call transgenic mice. So in a transgenic mouse, our DNA gene of interest, we insert it into the germline of the mouse, and we plant that, once it's in the germline, we create an embryo, and once we have an embryo, we can insert that embryo into a mouse, and then that offspring will have that transgenic piece of DNA. So it will have our DNA that we've manipulated. We sometimes do this. We can put human genes into a mouse when we do this. We can put, we can overexpress a gene, or we can put a mutated gene. So we take a natural gene, we can mutate it, put it into a mouse to then see what the outcome of having that mutated gene would be, and it helps us to study genetic diseases. We can also do what we call knockouts or knock-ins. A knockout is when we can, for our gene of interest, we can inactivate it. And we can do that by replacing the gene of interest with an artificial piece of DNA, or we can just disrupt the gene of interest with an artificial piece of, I'm sorry, not RNA, DNA. And then we used a very similar methodology to put then that knocked out gene into the germline of a mouse, and then excuse me, we implant the resulting embryo into a pseudopregnant mouse. All of the mice that are going to be born from this mouse then might have that gene of interest, and then we can crossbreed those mice. And when we crossbreed them, then we might produce a mouse that has our knockout gene of interest. So it will not express that gene, and so we can see what the absence of that gene and then corresponding protein has on the organism. A knock-in gene is when we add in a gene of interest. And so in all of these cases, when we create that plasmid, that original plasmid, generally we put markers for our gene of interest to know that it's going to be expressed. And so a lot of times our marker, since instead of dealing with bacteria, we're dealing with mice, our marker is going to be something to do with fur color.